Hello everyone, welcome to today's live broadcast. Visualization software creates results quicker and involves biologists. I'm Judy O'Rourke of Labroots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by Qlucore, a world leader in the development of bioinformatics software. Qlucore began in 2007 as a collaborative research project at Lund University, Sweden, supported by researchers at the Departments of Mathematics and Clinical Genetics in order to address the vast amount of high-dimensional data generated by microarray gene expression experiments. It was recognized that an interactive scientific software tool was needed to conceptualize the ideas evolving from the research collaboration. The Qlucore software takes full advantage of the most powerful pattern recognizer that exists, the human brain. Qlucore has clients in more than 25 countries, and most are from the life science and biotech industries. For more information, please visit www.qlucore.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button, lower left. I would now like to introduce today's speakers, Carl-Johan Ivarsson, President of Qlucore, and Tiwas Fleoretos, MD, PhD, Professor and Senior Consultant. Carl-Johan Ivarsson has more than 20 years experience in the software and telecommunication industries and broad experience from international business management and sales. He is one of Qlucore's founders and has been the company's president since 2007. He worked at Ericsson from 1998 to 2007, and during that period, Carl-Johan, among other tasks, built and managed the new product management unit at Ericsson Mobile Platforms in 2001 and headed Ericsson Mobile Platforms in China from 2004 to 2006. Dr. Fleoretos is a professor and senior consultant at the Department of Clinical Genetics, University and Regional Laboratories at Lund University, an elected member of several learned societies, associate editor of Genes, Chromosomes, and Cancer. He has published more than 100 scientific original research articles and reviews in international journals, serves as a reviewer for several national and international funding agencies, and is, apart from Qlucore, also a co-founder of Cantargia AB. I will now turn it over to Carl-Johan Ivarsson for his presentation.
Okay, thank you for the introduction. I will change here to my slide presentation, and I hope you can hear me in a good way. Okay. During the webinar, we will present how you can generate new scientific ideas and also free up bioinformatic resources. One part of the webinar will be a live demonstration of Qlucore Omics Explorer. We will also share examples on how Qlucore Omics Explorer is being used by our clients. If you study market reports, the overall figures for bioinformatics and data analysis, the annual growth is about 20%. This is the commercial aspect of the challenges that scientists and researchers are facing. I think it's fair to assume that we are all in agreement that the amount of data to be analyzed is growing very fast, and improved solutions are required to maximize the outcome of the generated data. Data analysis is currently a bottleneck in many organizations. To us at Qlucore and to our clients, it has been clear for several years that the solution to the ever-growing amount of data is not only faster computers, more hard disks, or new algorithms. All this is indeed needed, but it's not enough. The steep growth in number of analysts, statisticians, and bioinformaticians are one part of the solution. But again, more is required to improve the outcome of the vast amount of data and the knowledge buried within the data. When we meet clients, they commonly communicate that the lack of analysis resources blocks knowledge uptake, development, and slows projects down. Our piece of the solution is to enable data analysis closer to the data source. That is to involve the scientists and the researchers who design and perform the experiment or study. One significant aspect in making this possible is to provide a software tool that are fast and easy enough for non-expert users. Since these scientists execute a broad range of tasks and data analysis is only one of them, the scientists hence require software programs without deep learning curves and without extensive requirements on statistical background knowledge. New findings or improved results is in a broad sense what our clients strive for. There are many sources that point to that cooperation between groups and having the right competencies working on significant problems are fundamental aspects to achieve new findings and new results. To secure the involvement by scientists and better manage data analysis bottlenecks, we are using 3 key elements in the design of Qlucore Omics Explorer. Visualization is in the center. This gives visual and easy to understand feedback to the user during all steps of the analysis. It also enables the use of the best pattern recognition known to us, the human brain. A click and point user interface adds to making the program easy to use. No script and no requirements on writing code. Instead, the required functionality is only a few mouse clicks away. Speed. If data analysis and exploration shall be rewarding and available to scientists, the solution must be fast. Qlucore Omics Explorer is faster than all other solutions for advanced calculations, presenting updates instantly for the full range of data sets from small to large. This is also true for advanced and information-rich visualizations such as principal component analysis. At this point, we would like to make one clear statement. You should not mix up Qlucore Omics Explorer with pure visualization programs, which have very limited analysis capacity. These solutions normally only help you to make nice pictures of what you already know. Qlucore Omics Explorer will instead help you to find what you are looking for and drive your research further. Since we stand for visualization, what could be more relevant than to show you visually with a live demo what you want? But before the demonstration, I would like to share one example in form of a case study from one of our clients, and then also invite Stuart Theoretos, one of Qlucore's founders, to share his vision and one example on how he has been using Qlucore Omics Explorer. We start with a case study. The Center for Interdisciplinary Cardiovascular Sciences, CIC, was created in 2009 by the Brigham and Women's Hospital and the Cova Company. The center is located in Boston. The proteomics group at CIC, led by Dr. Sasha Singh, integrates the latest technologies in mass spectrometry and collaborates with the center's main research group. These groups studies focused on 4 major biological processes contributing to cardiovascular disease: macrophage biology, lipid metabolism, arterial and valvular calcification, and metabolic disorders such as type 2 diabetes. Their situation is that they have a lack of bioinformatics.
Slide to be on?
Hello?
Yes. What slide would you like to be on? I'm only seeing slide 1.
6.
Can you please move it in the studio?
Does it show? Can you see 6?
Yes, please continue.
The proteomics groups at 6, led by Dr. Sasha Singh, integrate the latest technologies in mass spectrometry and collaborate with the Center's main research group. These groups studies focus on 4 major biological processes contributing to cardiovascular disease: macrophage biology, lipid metabolism, arterial and valvular calcification, and metabolic disorders such as type 2 diabetes. Their situation is that they have a lack of bioinformaticians, and the majority of their staff are biologists, and their main challenge is to manage the research constraints. The solution they pursued was to implement Qlucore Omics Explorer for the biology. The outcome has been successful, and the deployment has delivered both improved understanding of the data for the biologists as well as freeing up the constrained information research. This case study and several others, including more details, are available at qlucore.com. And now I will hand over to Stuart Geretos, who will share with you some of his aspects.
Thank you, Carl-Johan. So I'm one of the founders of Qlucore, and I just wanted to share with you some ideas we had in the beginning when we started out to build this software. And I think it's fair to say that we wanted this software to analyze large-scale data sets to be easy, and it should only require basic knowledge. So I'm a medical doctor, biologist, and time does not permit me as a person or a leader of a research group to learn how to run scripts or write scripts. So the absolute requirement was that it should only require basic knowledge. We also wanted to bring the data back to the biologists and allow them to do initial data analysis and exploration. I think that's very critical if you want to build a successful project. That the data is not shared or that the data is not separated between biologists, medical doctors, or bioinformaticians. It should also drive creativity and generate new hypotheses. So it should be a highly interactive software. And it should, of course, then also facilitate the interaction with expert bioinformaticians and allow them to focus on more advanced analysis and free up time. So on the next slide, this is just an example that we used early on in 2010, where the Qlucore software was critical. So as you see, this resulted in a paper published in PNAS. And what we did in this study was that we took leukemic cells, or actually leukemic stem cells from patients directly. We did gene expression profiling by arrays and later on by RNA sequencing. And then we used Qlucore software and a feature that will be shown later by Carl-Johan Ivarsson, that you just look on subsets of your data. So in this case, we only looked for cell surface markers that were differentially expressed in the leukemic stem cells. And we can then later on validate that one of these receptors, called in this case IL-1RAP, was highly upregulated on stem cells. We could separate these leukemic stem cells, could generate antibodies that could then kill off the leukemic stem cells. So this is just one example where Qlucore has been critical for identifying a critical result in cancer biology. So with that, I hand the talk over to Carl-Johan Ivarsson again. Thank you.
Thank you to us. This brings us now close to the demonstration. And we will work with data from leukemia, and we will actually work with 2 different data sets. During the demonstration, we will generate new hypotheses by the identification of new subgroups, and also find discriminating variables for known annotation, and of course, visualize the results. Finally, we will compare our findings with public information and also limit the biological scope that we are addressing by using this. As I'm sure you understand, we're only showing a small part of the available functionality. The first step in data analysis is to import data, and we know that many users right over this and wonder, how do I import data and what types of data can be imported? Qlucore Omics Explorer uses generic algorithms and methods and do not make any assumptions on the type of data imported. So from that perspective, all types of data and of various sizes can be imported. Examples of data are gene expression, DNA methylation, proteomics, and so on. You can always use the wizard for import of any type of normalized data. For certain data types and instruments that are built-in, there are built-in workflows which includes normalization. Performance is very good, and also large data sets, with for instance 500 samples and 50,000 variables or larger, can be imported and analyzed with a normal computer. So I will now again try to share my desktop here with you. And I look like that it's working. So hopefully in front of you, you see now Qlucore Omics Explorer with a data set with 132 samples, and I'm slowly rotating a sample PCA plot with 132 samples. What we will do first here is to start with a data exploration. We will work to find structure and information in this data set. Before we do that, just give you one hint on how you interpret the PCA plot. I will show you more plot types later on, but two spheres, each representing a sample or a patient in this case, that are close to each other, have a similar expression pattern, and two spheres that are far from each other are very different. The data is gene expression, and the patients are children who are suffering from leukemia. We don't see any subgroups in the plot, and the reason for that is that a lot of the variables, currently the 22,282 process, are contributing only with noise. To handle that, we have to use the variance filtering, which is a built-in, very powerful functionality in Qlucore Omics Explorer, especially when it is combined with visualization. So this is really one of the unique features that I have talked about, that you get the instant visualization. I can move the slider slowly, and you can see how the data is changing. You can see in the PCA plot how data is splitting into two groups. This value here is called projection score, and it helps you to find the best visualization in a PCA plot. It shows you the highest level of information that is captured compared to random data. And somewhere around here, 0.43, we can see that there is a maximum. Here somewhere. And data is then clearly split into two groups. To capture that information, I will create a new annotation and give it a couple of values. I will color the plot, and then I will use my mouse tool over here, which controls a lot of the actions in the program. Make sure it is a multi there. And then now I've created a new annotation with the name new annotation. I changed that to Explore, and I have started to work and dig down into the structure of this data set. And as you can see, I maybe use my 10 clicks with the mouse, I have moved sliders and so on. And this is really how the program works and how easy it is. If I then would like to have a closer look to the blue part, I can just uncheck the yellow sample group here, and then I can adjust to see if I can find a good visualization. I can connect the nearest neighbors. I rotate a little bit, and you see there is much more structure in this data set than just to show you. I can classify it, and I start with my group like that. I can bring back my yellow group, and this is how easily it is to work with sub-selection. Now, just to prove that we haven't just invented strange things here and found non-relevant groups, I will in my right plot here have the samples colored according to my Explorer annotation, and in the second window here to the left, I will color the data according to an annotation called leukemia subtype, which is a classification of the samples based on other clinical parameters that the physicians that did the study originally have defined. And as you see, the yellow group we identified is equal to the orange group here, which is the CALL group, and the purple one is equal to the red one here, which is all the E2A PBX group. So we have found two biologically relevant groups just here in a few minutes using variance filtering, reaction score, and PCA. And if this now was a new data set, this was actually a very good way to get a feeling for the data set. If now this wouldn't have been matching towards my original hypothesis, which we can say the leukemia subtype annotation represents, I would have known that I potentially have some batch problems that I would need to take care of and so on. Now I would like to restart the analysis and have a different approach. So just to show you that, I will close down the data set. I will open it up again. So here we have it. It's the same data set, but I will do a little bit different approach. So the purpose now, my hypothesis, is that I would like to find the discriminating gene that can separate the CL-AML1 group from the others. So I start with coloring my data. Just to show you how easy it is to deselect groups, I will remove the CALL group. Then I need to set up my statistical test. Since I'm going to look for one group, the CL-AML1 group compared to the others, it's a two-group test I need. And I find that here, defined in the statistics dialog. I select my annotation, I select the CL-AML1 group, and then it's test. So, and then I can just move this slider and start to set the p-value cutoff to remove variables. But for the purpose of the demonstration, I will change to heat map so that you can see that there are more plot types, and I will have hierarchical clustering on that one, including and coloring according to this. I will also do one more change since the data set is from ProStet, but I would later like to compare with genes in other aspects. I changed the identifier to gene symbol. But then there is very common a problem that you need to solve. There are multiple ProStets matching to that gene symbol, so we need to collapse the data. So now I'm recalculating, and for all row in the data, each row in the data set will have now matched to more than one ProStet. We collapse them using an average method, and we can also change to gene symbol here on the axis. So we are done. Now we shall have a look on how many variables we have, and we see that here in the variable tab, and then I start to filter, and you get the interactivity again. I start to remove genes, and let's do really strong p-value cutoff, and we can clearly see that I'm separating out the blue group as we were trying to do, and here are all the others. And this list was then updated automatically, and that is the list of the genes that are currently taking part of the analysis, the ones that are best on separating CL-AML1 samples from the others. Here you see the p-value cutoff. Here is the corrected value, the false discovery rate. You can also work with fold change. So we are not only interested in the statistical significance, but also the effect. So let's add a fold change filter of 2. So now we have the genes that have fold 2 and this statistical cutoff. And then there are only 15, and maybe we should have a few more. So then I just easily change my slider here. Yeah, something like that. So we get both up-regulated genes and down-regulated genes. To capture this result, I make a copy, and then I have my list. So these are the ones that are separating the CL-AML group from the other. And you get all the elements of that list here, and you get the meta information about how it was done, what type of statistical test here, and that can all be saved. You can, of course, export images, you can export videos when you have PSA plots, and a lot of additional information. I should have added here so that you can clearly see the p-values. You can add the false discovery rate, and you have that column, and you have that in the data set. And then we just save that list, and everything is saved. So now I would like to move and to a second data set and to verify my results in that second data set. And I can do that by downloading, for instance, the data set from Gene Expression Omniva. And I just have to go here to File, Download, and View Data Set. Then I add my accession number. In this case, I already know it. But if you don't know exactly what you're looking for or would like to browse all available data sets, you get quick links here to Geo. But now I know. So now that I get a question which annotations I like. I have downloaded this data set before, so then it was a little bit quicker, but it's really quick also when you do it the first time. And this is a different leukemia data set with 190 samples. Then done by a different group or generated by a different Unicode group. It includes one annotation, which is characteristics, so the samples here are classified into different groups, so we can color according to that. We also need to handle the fact that the identifiers here are what I'm called ID ref, but we would like to have it on gene symbols so that we compare. And again, a few mouse clicks, and we have the data set prepared and organized according to gene symbols. If we just look at it, we can see that the blue samples here, which are the CLL, are sticking out quite significantly. And that was what we also saw in our other data sets after a little bit of filtering. That was exactly what happened. Now, let's see how this second data set looks if I use the result from my first data set. And again, how difficult is that? It's only a mouse click away. So now I use the base of the 34 genes from my first data. Actually, there are 29 matches of those genes in this data set. And what happens? We step clearly. The PSA plot shows that those 34 genes are separating out these yellow samples over here, and they are the CL-AML1. So we generated a list of the best discriminating genes for the CL-AML1 samples or the group in one data set. The use that list of discriminating variables and visualize how it looked in a PSA plot in a second data set, and we have verified and looked at the biological significance. We can then further refine the analysis by opening, let's see here, we can open a variable list. Here we go. We can take kinases. So this is a list of genes that are classified as kinases, and we see that they have 146 matches. So now if I instead do like that, and we go back to my first data set. Sorry, like that, a little bit less interesting. What are we doing now? Now we are looking for the genes that are discriminating the TL-AML1 group from the other group, but that also are classified as kinases. And then we have that result here again. So that is how easy it is to restrict and work only with subsets of your variables, and do that with a lot of relevant context, depending on your research area, what pathways you're interested in, what others have published, and so on. That is what I plan to show you here, a very, very quick brief showing 2 plot times and giving you a feeling for the flexibility of Qlucore Omics Explorer. And let's see if we can move back to the slide. When you have seen the short demo, and we would like to show you how you can implement this in your own organization. The first example is within academia. The scope of analysis within academia is normally very broad, and both exploration and analysis is required. The license type that fits that very well is a floating license, since it will give a whole group access to the program, but with limited cost. Since data comes from many sources and there are normally no specialist support, the scientists need support from a program to be able to import data, and that is easily handled in Qlucore, and the width of it is a very flexible way of getting data into the program. As I mentioned, easy download of gene expression omnibus data, and also a built-in gene set enrichment analysis workbench, which makes it very easy. There is a broad range of statistical tools. I only showed you the 2-group comparison and fold change, but there is complete ANOVA framework and with included batch corrections. And then you can export your results for publications and with relevant meta information to keep track of your work. If you look at the example from the pharma industry, then it's more used for exploration and hypothesis generation compared to doing statistical tests. Since normally within pharma there are bioinformaticians and statisticians that can help the scientists with statistical tests. That is also relevant for the data import aspect, as there is normally more support. One very straightforward way is to use pre-processed data instead, and then we have a format for that. We call that G-data, so scientists can get data from central storage within the organization. But then access to public data can be very valuable, and for sharing and keeping track of your work, we have a special functionality for log points where you freeze the state of the program and then easily can go back. Exports and so on are similar and easy to use as I showed you. Thanks for watching, and we would like to start to summarize what Qlucore Omics Explorer can bring to your organization. In short, It enables creative analysis by maximizing scientific involvement and optimizing the use of research. The signature of Qlucore Omics Explorer is that it is for everyone. All actions and selections are done with checkboxes, sliders, and a mouse. It supports explorative analysis based on visualization and is much more powerful than a traditional reporting tool. The user is always in full control through the instant visualization and also the easy-to-use click and point interface. The program can be used by both experts working with it every day and by users who also fulfill many other tasks. This is possible since it's easy to learn and easy to remember. Then the question is, will this work in your organization? Of course. One proof point is that the list of more than 300 publications at Qlucore.com, they are all published by our clients and referring to the use of Qlucore Omics Explorer. But it's always best to try by yourself. So we have a free trial. You can download it at Qlucore.com. We will give you full support during the trial. And if you have any questions now or later, feel free to ask them. I know that we already have some questions, and let's see what our host is going to help us with here.
Yes. Thank you both for sharing that information with us. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many questions as we can. And our first question is, does Qlucore Omics Explorer require any special hardware?
No, it doesn't really. You can run it on any Mac or any Windows-based computer, and it is very resource efficient. So you don't have to have the toughest hardware requirements. It really works very well.
Okay. Can more than one user in my organization use the trial version?
Absolutely. The trial version is free for download, so if you're 5 in the organization, each of you can download, no problem.
Does it work on RNA-seq data?
Absolutely. There is a direct import functionality for so-called LAMP files, so that you can import it directly, and it is normalized, and you're up and running, analyzing your data within minutes.
How can it be so fast?
Yeah, there are many reasons for that. One is that we have spent a lot of effort in writing efficient code and selecting the algorithms very clearly. And I think also it's best experience how well the speed helps you when you try it yourself.
How often do you release updates and add new functionality?
Yeah, we normally do one release every year, and then we add new functionality.
I know it is possible to use Qlucore Omics software to screen your database for genes grouped into known pathways, such as autophagy or protein synthesis. But how do I do this?
Okay, that is typically what you can use the inbuilt gene set enrichment analysis workbench for, which I didn't show you. But download the program and try, and we will assist you. There is also documents around this for how to analyze pathway documents on the documentation part of Qlucore.com.
What is used already by reproduction groups to identify new potential targets in human infertility?
Yeah, I don't know that on the top of my head here. please have a look at all the references that we have on Qlucore.com.
Okay. How about proteomic data from mass spectrometric analysis?
Okay, excellent question. Again, here, thank you all for the questions. It works perfectly well on proteomic data. We have a lot of customers who do that. As I mentioned, the case study here from 6, they are working with the mass spec data. So, and there are many other examples of that too.
If there are no more questions, I would like to once again thank Carl-Johan Ivarsson and Dr. Fiorettos for their presentation. Do you have any final comments for us today?
Yes, thank you very much, all of you, for watching. We understand that there has been a lot of technical challenges, but also seen that many of you hang in there. Thank you very much for that. Visualization is really going to help you in your research, and the best way is to try it. You will get the instant visualization and support in doing and finding new results in your hand, getting in touch with your data. Thank you.
Yes. And you're speaking for both of you then, I would say. I would also like to thank our sponsor, Qlucore, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through June 1st, 2016. You'll receive an email from Labroots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.